Increased accessibility and improvements in induced pluripotent stem cell technology have allowed complex human biology to be reproduced in vitro at previously infeasible scales. iPSCs are now commonly used to improve disease models, accelerate drug discovery, and screen potential toxic compounds. Accurate characterization of iPSC-derived cells requires functional assays to determine cellular phenotypes and assess differences in disease models or in response to drug or compound treatment. Axion Biosystems Maestro Pro and Edge multi-well microelectrode array platforms offer a solution by providing non-invasive, label-free electrophysiology to simply and accurately record functional activity from electrically active cells. Here we use the Maestro MEA platform to record functional electrical activity from cardiomyocytes and neurons and present a multiplexed assay to assess both function and structural integrity of these populations. When using functional activity to assess the efficacy or toxicity of a compound, it's important to consider cell health in addition to functional effects. Traditionally, this required a separate viability or similar endpoint assay. With the Maestro, impedance technology is used to assess the structural integrity of the cells while simultaneously recording functional electrical activity. Cardiac field potential recording is a sensitive and validated method to detect acute and chronic toxicity. To distinguish compounds that produce a cardiotoxic effect from compounds producing a cytotoxic effect, we use the MEA viability module to track both function and structural integrity 6 days after compound treatment. Functional activity was reduced as early as 30 minutes to 12 hours after treatment for all 3 compounds tested. Structural toxicity, however, was only induced by doxorubicin and pentamidine, with doxorubicin causing cell death within 24 hours and pentamidine causing cell death gradually over 6 days, demonstrating the importance of measuring both function and structural integrity. For neurons, the Maestro provides a comprehensive assessment of neural activity, neural connectivity, and structural integrity. To assess neurotoxicity, rodent cortical neurons were seeded and matured on 48-well MEA plates, then treated with 12 potential proconvulsant or anti-epileptic compounds. Network burst intensity was measured for each compound, increasing for proconvulsant compounds and decreasing for anti-epileptic compounds. Finally, we assess viability in a similar manner to the cardiomyocytes using the MEA viability module. For compounds which decrease or shut down neural activity, such as anti-epileptics, MEA viability allows for non-invasive assessment of cell health to determine if these compounds are cytotoxic or only affecting functional activity. Here we've demonstrated how the Maestro MEA platform can easily be used to simultaneously evaluate both structural integrity and function of iPSC-derived neurons and cardiomyocytes. For more information, please visit poster 699 and booth 715 on site at ISSCR.